Hello again, everybody. Welcome back. Today we're here to talk about energy pathways. So in our last videos, we talked about energy that's required by living systems, how enzymes are used in order to reduce the energy that's used to conduct chemical reactions. And now we're going to talk about how our bodies actually create that energy. And specifically, we're talking about the process of cellular respiration. So, as you probably noticed, right up here, we've got a picture of one of my favorite meals of all time, a cheeseburger with fries and a milkshake. And the reason I put that picture up here is just to kind of remind you that, when we eat these meals, there's certain things that our bodies get from those different foods. For example, the milkshake is a great source of lipids. The burger itself is a great source of protein. And then the fries and the bread and some other things are great sources of carbohydrates. And that's really what we're going to be interested in here today, are carbohydrates. And specifically, we're going to be looking at glucose, which is the monomer of carbohydrates. So, when we talk about energy, obviously we've talked about ATP and how our cells use ATP to get energy, but ultimately we need glucose in order to make that ATP. So this is a chemical formula you should be familiar with by the end of this particular unit. So over here on the left, we've got C6H12O6, which is the chemical formula for glucose, plus oxygen gives us carbon dioxide, water, and free energy. And by free energy, we mean ATP, right? And this process takes place in several different steps. And we're going to go back and revisit each of these steps in order with a little bit of detail for each. So the first of those is what we call glycolysis. And this is the process by which glucose gets broken down into something called pyruvate. And then glycolysis ultimately leads to cellular respiration. So this is the process where we actually are taking the product of glycolysis, so this pyruvate, and we're using oxygen plus pyruvate and a few other things, and that's ultimately going to give us ATP. That's our ultimate goal here, is to create energy for the cells. And lastly, we'll also talk about something called fermentation, which is how our bodies process energy when there's a lack of oxygen. So that's what we refer to as anaerobic conditions, whereas cellular respiration is in the presence of oxygen, making it an aerobic process. So let's start off by refreshing our memory on something called redox reactions, because cellular respiration ultimately is just one big redox reaction. And there's this mnemonic that can help you remember what those words mean, using oil rig as our example. So if you remember, the oil part of it means that oxidation is losing, and the rig part means reduction is gaining. And in both cases, we're talking about electrons. So when something is oxidized, it loses electrons. When something is reduced, it gains electrons. I know those terms are they don't really fit with what's going on here. How do you reduce when you're gaining something? But the way I always remembered it was electrons are negatively charged, so you're reducing the overall charge of this molecule when you add electrons. So in essence, you are reducing the charge. It's one way to maybe help you remember all of this. But in this case, we're talking about, we're starting with glucose, and ultimately our electrons, I'm going to use a little E- to symbolize electrons, in this case are going to oxygen. And so at the end of the reaction, our glucose is going to end up becoming carbon dioxide. It's going to be oxidized into carbon dioxide, and oxygen is going to be reduced in order to make water. So that loss of electrons converts our glucose into CO2 and converts our oxygen into water, and then the energy that's exchanged in the path in the middle there is used to make ATP. So, how are those electrons transported from one place to the other? If you remember anything from chemistry, electrons are really, really high energy, and if they're not connected to something, they're just going to fly off into space and they're not going to do us any good. So we need molecules that are going to shuttle these electrons from one place to the other. In this reaction, the key electron carrier is this NAD+. So NAD+, you don't need to remember this, but that name stands for nicotinamide adenine dinucleotide. You don't need to remember that long name. What you do need to remember is that NAD+ is something called a coenzyme. And so in this case, NAD+, this is the oxidized form. And then if you add electrons, so here we're going to add 2 electrons and 1 hydrogen. So that hydrogen is going to add to this and make it NADH2+, and then the 2 electrons are going to cancel that out, and we end up with NADH. So you can see the H is added right here and ends up in this spot, and now we're carrying these 2 electrons as well. So this is our reduced form over here. And then this NADH ultimately can take the electrons that it gathered and it can deliver them somewhere else. It can be oxidized and it returns back to NAD+. So just like ATP is constantly being recycled between ADP and ATP, NAD+ is constantly being recycled to NADH and then back and around and around and around. There's also another one that you're going to see here called FAD, and that's its oxidized form, and the reduced form is FADH2. But we'll get to that in just a little bit. Okay, so in aerobic conditions, we're gonna that's the real determiner of how our bodies process energies, whether there's oxygen available or not. In aerobic conditions, there's a certain path that's followed. So we always start with glycolysis. This is what's called the universal pathway. And it's called that because even prokaryotic cells utilize glycolysis. If you remember, lysis means to break. Glyco refers to glucose. So you're literally breaking glucose open. And you're creating something called pyruvate. If oxygen is present, we're going to continue this process. If oxygen's not present, we'll show you in a second here what happens. In the presence of oxygen, our next step is something called pyruvate oxidation, followed by citric acid cycle, followed by electron transport and ATP synthesis. And at the end, we've got our waste products, which are carbon dioxide and water. We're going to cover each of these steps in a little bit more detail, so don't worry if this is still kind of confusing to you. If oxygen's not present, notice we're still going to start with glycolysis. It's going to take the glucose and it's going to break it down into pyruvate, but then in the absence of oxygen, we're going to skip all those other steps and we're going to go through something called fermentation. And fermentation leads to the production of either lactate or alcohol. In our cells, we produce lactate. Yeast and some bacteria and things like that produce alcohol during fermentation. But we'll get to fermentation in our next video. So let's start with glycolysis. This is where energy production always begins. Glycolysis is a multi-step process, and I will say at the beginning here that all of these processes, you don't need to remember all the individual little steps. It's more just the big picture. So first thing about glycolysis, where does it take place? Well, it happens in the cytoplasm. Remember, this is the universal pathway. It happens in it happens in eukaryotic cells, prokaryotic cells. All cells use glycolysis. So it doesn't rely on organelles in order to happen. It just happens right in the cytoplasm. And there's 2 main phases to glycolysis. There's the energy investment phase, or energy investing phase, and then there's the energy harvesting phase. So what I'm showing you here, this is the energy investing phase. So we're starting with glucose, that's this molecule right here. And in the first 3 steps, notice we're taking ATP and we're converting it to ADP. So these ATP molecules are losing a phosphate group that's going to be added to our glucose. And what you end up with is an intermediate molecule. Again, you don't need to remember this name, but this is fructose 1,6-biphosphate. So this is fructose, and on the number 1 and the number 6 carbons, we have phosphate groups added that came from the ATP. At this point, now we're going from the energy investing phase to the energy harvesting phase. And one thing I want you to pay attention to here is notice that this splits. And now this fructose 1,6-biphosphate during this next process is going to be split into two 3-carbon molecules. And so you're going to see kind of 2 chains of the same reaction happening over and over again. And here we are in the energy investment phase, or I'm sorry, the energy harvesting phase. So now you'll see that we have ADP that's being used to make 1, 2, 3, 4 ATP molecules. So we started by investing 2. So we used 2 ATP, but we gained 4 ATP. So our net gain is 2 ATP. That's our net gain. And then you also notice that up here, we have 2 molecules of NAD plus that were reduced. They gained electrons to become NADH. So what's the take home here? In glucose, we start with glucose, ADP, and inorganic phosphate, and NAD+. We end with 2 molecules of pyruvate, so this glucose directly becomes that. These become ATP. I guess we could have said there's 2 of those, and there's 2 of these that ultimately become 2 NADH. And again, all of this happens in the cytoplasm. So this is the first step in harvesting energy. We're taking glucose and we're breaking it into 2 3-carbon molecules called pyruvate. They're sometimes also called pyruvic acid. So either one of those is the same thing. Now, if oxygen is present, the next step in this process is what we call pyruvate oxidation. So we're going to take that pyruvate, we're going to oxidize it, which means we're going to take electrons away, right? So here's our pyruvate. And notice we've got the cytosol here and the mitochondria. So now we're moving into the mitochondria. I always thought of it as mitochondria as it's kind of like the club, and the cover charge to get in is oxygen. So the only way this happens is if we have oxygen present. So the oxidation part happens right here. So electrons from pyruvate are removed, added to NAD plus in order to make NADH. In the process, carbon dioxide is lost, and then a coenzyme called coenzyme A is added, and ultimately what we end up with is something called acetyl-CoA. And this is what we need for our next step in the process, the acetyl-CoA. So again, summing everything up here, in pyruvate oxidation, we start with our 2 pyruvate molecules that came from glycolysis. We have 2 coenzyme A's and 2 NAD+. And here our pyruvate ultimately is going to lead to our CO2 as well as our acetyl-CoA. Our coenzyme A is going to help make up this acetyl-CoA, and then the NAD+ is going to become NADH. And the next step is what we call the citric acid cycle. It's also sometimes referred to as the Krebs cycle, but it's more commonly known as the citric acid cycle because of this intermediate right here, citrate, which is also called citric acid. Now, on the surface, this looks like a very complex process, and it is. This is kind of an entire semester of biochemistry just summed up in this one diagram. But there's really only certain things that we are concerned with here. So remember, we started with a pyruvate. We started with 2 pyruvate molecules, and that became 2 acetyl-CoA molecules. So this whole thing is happening twice for every molecule of glucose. The key things that I want you to pay attention to are right here, 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 and here. And so you'll notice at this step, we are oxidizing our intermediates and we're reducing NAD plus into NADH. In the process, we're losing carbon dioxide. So every glucose, there's 2 of all of these happening. Same thing happens again here. 2 NAD plus become 2 NADH, and 2 more carbon dioxide molecules are removed. I'm going to come back to this one here at the bottom. Right here, now we've got 2 FADs that become 2 FADH, which is again just another electron carrier. And lastly, we've got one more step where another 2 NADH molecules, or 2 NAD plus, become 2 NADH. And you'll notice that all of these intermediates keep recycling over and over and over again, which is why it's called the citric acid cycle. What comes out of this is right here at the bottom, we have GDP that is combined with inorganic phosphate to make GTP. And then ultimately, the energy from GTP is used to make ATP. And again, we get 2 of them for every molecule of glucose. So again, let's sum this up and make it a little bit easier. In the citric acid cycle, we're starting with 2 molecules of acetyl-CoA, 2 ADP, 6 NAD+, and 2 FAD, and these are our products. So our acetyl-CoA ultimately becomes carbon dioxide. These become ATP. This goes right here, and FAD becomes FADH2. Now, I know we've gone kind of quickly, so let's kind of summarize everything up to this point, and that's where we're going to end for today. So, in our cells, we start with glucose. We also require ADP, NAD+, and FAD2, or FAD. At the end of this process, what we end up with is CO2, which comes directly from our glucose, ATP, which comes directly from our ADP and inorganic phosphate, NADH comes from the NAD+, and the FADH2 comes from the FAD. What all of these are used for, we will talk about in our next video. The one I can tell you about, this is just simply a waste product. That's something we breathe out. ATP, we know what ATP is used for. It's used for energy in cells. But then what are these guys, these electron carriers, what are they going to be used for? That's going to be answered in our next video. So we'll see you next time.